Hello all, my name's Heather Madsen. This is a biotech lecture exploring the potential of menstrual-derived stem cell treatments. So today you'll come away from this lesson learning about the potential use of menstrual-derived stem cells in treating and reversing premature ovarian failure in women under the age of 40, specifically those who underwent chemotherapy. You'll learn that menstrual-derived stem cells are pluripotent, giving them a wide range of differentiating abilities, and you'll also learn that menstrual-derived stem cells are not as ethically controversial as embryonic stem cells, which makes them more practical in stem cell therapy. You'll leave today knowing what stem cells are, what premature ovarian failure is, and the key differences between bone marrow-derived stem cells and menstrual-derived stem cells. So this is just a short TED-Ed video about the basics of stem cell therapy.
Imagine two people are listening to music. What are the odds that they are listening to the exact same playlist? Probably pretty low. After all, everyone has very different tastes in music. Now, what are the odds that your body will need the exact same medical care and treatment as another person's body? Even lower. As we go through our lives, each of us will have very different needs for our own health care. Scientists and doctors are constantly researching ways to make medicine more personalized. One way they are doing this is by researching stem cells. Stem cells are cells that are undifferentiated, meaning they do not have a specific job or function. While skin cells protect your body, muscle cells contract, and nerve cells send signals, stem cells do not have any specific structures or functions. Stem cells do have the potential to become all other kinds of cells in your body. Your body uses stem cells to replace worn-out cells when they die. For example, you completely replace the lining of your intestines every 4 days. Stem cells beneath the lining of your intestines replace these cells as they wear out. Scientists hope that stem cells could be used to create a very special kind of personalized medicine in which we could replace your own body parts with, well, your own body parts. Stem cell researchers are working hard to find ways in which to use stem cells to create new tissue to replace the parts of organs that are damaged by injury or disease. Using stem cells to replace damaged bodily tissue is called regenerative medicine. For example, scientists currently use stem cells to treat patients with blood diseases such as leukemia. Leukemia is a form of cancer that affects your bone marrow. Bone marrow is the spongy tissue inside your bones where your blood cells are created. In leukemia, some of the cells inside your bone marrow grow uncontrollably, crowding out the healthy stem cells that form your blood cells. Some leukemia patients can receive a stem cell transplant. These new stem cells will create the blood cells needed by the patient's body. There are actually multiple kinds of stem cells that scientists can use for medical treatments and research. Adult stem cells or tissue-specific stem cells are found in small numbers in most of your body's tissues. Tissue-specific stem cells replace the existing cells in your organs as they wear out and die. Embryonic stem cells are created from leftover embryos that are willingly donated by patients from fertility clinics. Unlike tissue-specific stem cells, embryonic stem cells are pluripotent. This means that they can be grown into any kind of tissue in the body. A third kind of stem cells is called induced pluripotent stem cells. These are regular skin, fat, liver, or other cells that scientists have changed to behave like embryonic stem cells. Like embryonic stem cells, they too can become any kind of cell in the body. While scientists and doctors hope to use all of these kinds of stem cells to create new tissue to heal your body, they can also use stem cells to help understand how the body works. Scientists can watch stem cells develop into tissue to understand the mechanisms that the body uses to create new tissue in a controlled and regulated way. Scientists hope that with more research, they can not only develop specialized medicine that is specific to your body, but also better understand how your body functions, both when it's healthy and when it's not.
So now that you've learned just a little bit about stem cells and how they can be used in regenerative medicine, take a moment, pause this video, and ask yourself these questions. What are stem cells? What is the difference between totipotent, pluripotent, and multipotent stem cells? What kind of stem cells would you most likely find in an adult? And what are induced pluripotent stem cells? So hopefully you were able to answer at least a couple of these. Some of them are more challenging, but basically stem cells are blank slate cells. They are undifferentiated cells. Basically, your cells can be differentiated by the expression or repression of different alleles coding for different proteins. And those proteins code for different traits and specialize your cells. So when those different proteins are present, that tells you what type of cell that is. And basically the variations of stem cells have very, very few of those proteins, those alleles being expressed or repressed. So they can go in all these different directions. Once cells are differentiated, however, they cannot undifferentiate, naturally at least. And that's where induced pluripotent stem cells come into the picture. Basically, in the lab, scientists can change the expression and repression of different alleles so that the array of proteins present in the cell goes back to being a stem cell. And that gives those cells the ability to differentiate into any different type of cell throughout the body except the umbilical cord and the placenta, which brings us to the different types of stem cells. So totipotent stem cells are the most powerful stem cells you'll find anywhere. So totipotent stem cells are what you see when the zygote forms right after conception. Those cells can differentiate into any different types of tissue throughout the human body, including the placenta and umbilical cord. The pluripotent stem cells are a step down from that. They can differentiate into any type of tissue cells except that placenta and the umbilical cord. Finally, multipotent stem cells or adult stem cells can differentiate into an array of different cells specialized to certain tissues. So those are way more specific than pluripotent and totipotent stem cells, which restricts their application in a clinical setting, which is why pluripotent stem cells and embryonic stem cells are of such great value. But they're really controversial because of different ideas about when life begins and how embryos are being sacrificed to clinical research makes some people uncomfortable, while other people see it as common sense. So now we're going to take a turn and focus on cancer. Cancer basically is the uncontrolled growth of abnormal malignant cells. It is treatable and can be curable if it's caught early enough, but it also can be deadly. So normally the process of mitosis or asexual cell reproduction occurs only when cells die and they need to be replaced. Our cells are density dependent and they're anchorage dependent. They need to be attached to something. Otherwise, when our cells break off and go into the bloodstream, then they would naturally divide and they could clot and cause serious issues in our body. That's why our cells are supposed to be anchorage dependent. And then of course density dependent can also lead to tumors. So we have natural systems set in place to prevent unnecessary cell growth. There are different checks along the process of mitosis that prevent mutated DNA from being passed on to daughter cells. However, when there are mutations to the to specific areas of the genome that can result, specifically the tumor repressor genes, that can result in proto-oncogenes sending too many signals to replicate cells, and that can lead to cancer. So oftentimes cancer is treated with chemotherapy, can be treated with surgery if caught early enough again, but oftentimes, just to make sure that those really fast-growing cells are killed, chemotherapy is used, because that specifically targets cells that regenerate really quickly. And that's why you'll see various consequences on other parts of the body, like loss of hair, ulcers, because those some healthy cells that we need also regenerate really, really quickly. And those cells are equally targeted by chemotherapy and other cancer treatments. So we see that chemotherapy causing premature ovarian failure. And premature ovarian failure is basically irregular ovulation, premature menopause, or infertility experienced by women under the age of 40. And as I described, it can be caused by chemotherapy. Basically, it's when the cells lining the ovaries become infertile by increased rates of apoptosis or cell suicide, so that the ovaries cannot foster life. Besides chemotherapy, premature ovarian failure can also be caused by radiotherapy, environmental factors, or it can be inherited through a genetic mutation on the X chromosome. And here on the left of my screen, you'll see that X chromosome and the mutation. So I want you to pause the video and ask yourself, what are some possible treatments for premature ovarian failure? So this is probably a stretch, but did any of you guys come up with the question, what if doctors could combat premature ovarian failure by restoring the health of female ovaries through the process of inducing functional pluripotent stem cells isolated from the menstrual blood of healthy young women? It's a mouthful, but it can be done, theoretically. Using stem cell therapy, doctors have successfully restored fertility in women suffering from premature ovarian failure by using the most traditional bone marrow-derived stem cells. Bone marrow-derived stem cells were discovered in 1949, which has given them a significant head start in stem cell therapy over the more recently discovered menstrual-derived stem cells. They were discovered in 2007, and since that's really a blink of an eye within the scientific community, they have not ever been used on humans. However, a number of research projects have been done to explore the potential of menstrual-derived stem cells on mice, and some of those experiments have been done to explore the ability of menstrual-derived stem cells to reverse premature ovarian failure. One thing that's super promising about pluripotent menstrual-derived stem cells is that they do have a wider range of differentiation than multipotent stem cells such as bone marrow-derived stem cells, so they could be more effective at treating premature ovarian failure, just because they would be able to differentiate it into a wider range of cells that may be necessary in the ovaries, and they would be less likely to cause immune rejection, which we'll discuss in a moment. So because pluripotent menstrual-derived stem cells are able to differentiate into any cells throughout the human body besides cells comprising the placenta and the umbilical cord, menstrual-derived stem cells have incredibly low immunogenicity. This means it's far less likely for a stem cell recipient to have an immune response to and ultimately reject a menstrual-derived stem cell treatment, much more so than if that recipient received a bone marrow-derived stem cell treatment. As a result, menstrual-derived stem cell treatments would theoretically be safer and thus more effective than bone marrow treatments. And that safety extends beyond protecting the menstrual-derived stem cell treatment recipient. Donating these stem cells is super safe and easy, unlike bone marrow extractions that increase one's risk of deadly infections for months or even years after donating, require greater dependence on antibiotics, which in turn powers the evolution of antibiotic-resistant strains of bacteria, and leave the donor lethargic for days. Women who wish to donate menstrual-derived stem cells need only collect 10 milliliters of their menstrual blood in sterile diva cups during the first few days of ovulation for each month. The ease of donating increases the number of people willing to contribute their genetic diversity to this fight against premature ovarian failure. This increased genetic diversity, in turn, increases potential similarities between donors and recipients to reduce the risk of immune response yet more. Hopefully now you all see the real promise of menstrual-derived stem cell treatments, but now that I've explained how they're advantageous over traditional bone marrow treatments, I want you guys to pause and consider how menstrual-derived stem cells are isolated. So once you've had a moment, I'm just going to discuss the procedure. So only women ages 22 to 30 who consent to this procedure will collect and donate 10 milliliters of menstrual blood in sterile diva cups. That blood is then transferred into falcon tubes containing antibiotics to kill off any pathogens, bacteria that we do not want to culture and incubate. Because the next step is suspending, culturing, and incubating that blood at 37 degrees Celsius. Then mononuclear cells are coaxed to divide until 70% confluency is achieved. Finally, to induce the differentiation of isolated menstrual-derived stem cells into the epidermal lineage, the isolated cells are co-cultured with keratinocytes. This process renders isolated menstrual-derived stem cells for stem cell treatments. With those isolated stem cells, a number of independent labs have conducted research on mice to test the regenerative potential of menstrual-derived stem cells. One lab chose to measure the effectiveness of menstrual-derived stem cell treatment by measuring the rates of apoptosis, or cell suicide, and of cell proliferation in the ovaries of female mice. This required a control group of 10 mice who did not receive any treatment, a group of 20 mice injected with a combination of amino acids and minerals meant to repair premature ovarian failure, and a group of 20 mice injected with menstrual-derived stem cells. In one trial, the mice were sacrificed after 7 days, and in another after 21. Examination of the ovaries indicated that mice with induced premature ovarian failure suffered from weight loss caused by high rates of apoptosis, while the mice treated with menstrual-derived stem cells experienced high rates of fibroblast growth factor 2 secretion, which is a protein vital to proper cell regulation and growth. They also experienced inhibition of apoptosis and a greater presence of healthy follicles, including pre-ovulatory follicles. The menstrual-derived stem cell treatment thus significantly ameliorated ovarian damage, promoting tissue restoration to combat infertility by establishing a healthy microenvironment. In another experiment that utilized a more severe medium, hydrogen peroxide, to cause ovarian damage, 88 female mice were selected at random. 22 went unadulterated as the control group, while the other 66 were exposed to 10% hydrogen peroxide. The premature ovarian failure group was then further subdivided into a model group of 22 mice, a group of mice treated with synthetic estrogen. That group consisted of 22 mice as well, and finally a menstrual-derived stem cell treated group of 22 mice. After 7 days, 3 mice from each of these groups were randomly selected to be killed to observe their ovaries, comparing changes in hormone levels, ovarian index fertility rate, ovarian morphology, and ostrus cycle. This was repeated after 14 days. Then 10 mice from each group were paired with fertile male mice to measure fertility. Surprisingly, the premature ovarian failure group treated with menstrual-derived stem cells experienced 100% reproduction, not passing on any abnormalities, including infertility, to their offspring. The synthetic estrogen group yielded a fertility rate of 0%, the model 40%, and the control 80%. These results, in conjunction with molecular observations, indicate that menstrual-derived stem cells greatly promote ovarian failure repair, even restoring fertility in females suffering from premature ovarian failure by fostering proper cell and hormone regulation while inhibiting apoptosis. Now, the scientific community is still years away from any sort of human experimentation using menstrual-derived stem cells, but if the scientific community already condoned clinical menstrual-derived stem cell use, what would be the best method of delivering treatment? Please pause this video and consider for a moment the various ways of delivering menstrual-derived stem cells. So, low immunogenicity reduces the risk of rejection after administering treatment via intraovarian injection. In the experiments on mice, the stem cells were injected into the tail vein, demonstrating no signs of ovarian migration or differentiation. However, the treatment still restored fertility by fostering a healthy microenvironment. So that begs the question whether the location is really that important or whether it's more the microenvironment that fosters the restoration. That's why a human trial is really so important to better understanding the correlation between the location of injection and the fertility restoration of the treatment. Injecting menstrual-derived stem cell treatments into the bloodstream near the ovaries could be just as effective at reversing premature ovarian failure in women as injecting treatments directly into the ovaries. Such may reduce the risk of infection, which would be a demonstrable positive. So for that information, it would be wise to look to the history of treatments with bone marrow-derived stem cells, although there are still variations because those bone marrow-derived stem cells are have much less capability to differentiate and therefore have a limited treatment in comparison to menstrual-derived stem cells. While one could easily take the proportions of mice, the ovaries of female mice, and amplify the concentration of menstrual-derived stem cell treatment to accommodate the greater size of female ovaries, the physiology of women is greatly different from the physiology of female mice. So again, that just keeps coming back to the importance of understanding how menstrual-derived stem cells affect humans, and that really requires a human trial. So while menstrual-derived stem cells are self-replicating, there still must be an ample number of stem cells to enable restoration of fertility through cell growth, recovery, and repair. So again, just the best way to determine treatment dosage would be to conduct experimental trials on human subjects, of course with their consent. But since progress comes so slowly, it's really important to look to that dosage of bone marrow-derived stem cells. So, in conclusion, stem cell research is rapidly providing hope in the realm of regenerative therapy. Though much research remains to be done, menstrual-derived stem cells are perhaps the most promising due to their many advantages over the more common bone marrow-derived stem cells, including high proliferation rate, pluripotency, reliability, low immunogenicity, and lack of ethical gray area, because those menstrual-derived stem cells are not controversially seen as a form of life. They offer a wide range of clinical applications similar to those offered by embryonic stem cells, but without all that ethical controversy. Menstrual-derived stem cells offer incredible potential to women suffering from chemo-induced infertility due to their ability to foster healthy microenvironments by interacting with fibroblast growth factor receptors and inhibiting detrimental rates of apoptosis or cell suicide. With further human trials, menstrual-derived stem cells may come to overshadow the world of stem cell therapy. Thank you so much for sticking with me. Just to check your understanding, ask yourself to define stem cells, describe premature ovarian failure, and list a few important differences between bone marrow-derived stem cells and menstrual-derived stem cells. How do those differences impact their applications in stem cell therapy? Thank you so much. I hope you enjoyed this and learned a lot. Feel free to forward this video on to other people who you think are interested. And again, stay safe and healthy.